Hello, good afternoon, everyone. Today we are going to start our new topic, that is oogenesis. Oogenesis is what? It is a process of formation and maturation of ova, and where it is formed and matured. It is ovary, which is a site of formation and maturation of ovum, right? Ovary is a site of formation and maturation. Now, large germinal cells with bigger nucleus are called as primordial germ cells. These primordial germ cells are the precursor of all the follicles, all the ovum of female. Ovum are present inside the follicles, right? It involves production of haploid nucleus as well as acquisition of food reserves and preliminary organization of cytoplasm. Look at the female reproductive system. It consists of fallopian tube, ovary, uterus, and vagina. So what is the site of synthesis of yolk? It is also ovary and liver. And what is the site of oogenesis? It is the ovary. Look at the ultrastructure of ovary. It consists of blood vessels, nerves, some of the immature follicles, some of the mature follicles. See, it is surrounded by layer. It divided into two parts, that is ovarian cortex and ovarian medulla, right? So this is the site of oogenesis, that is ovary, a mature follicles which formed inside the ovary. What happens? The primordial follicles, meiosis 1 has been arrested in the primary oocyte, inside it, and around the oocyte, zona pellucida starts developing, and after that, a fluid-filled cavity that is known as antrum starts develop, and after that, mature follicles is formed, and after that, it ruptures from the ovarian wall is ruptured and ovum is released into the abdominal segments, right? Now, oogenesis, it divides into three parts, or it consists of three stages. First is multiplication phase, in which cells multiply, and second is growth phase, in which no multiplication takes place, only growth is takes place, increase in the size takes place, right? In the third phase, maturation phase, the size takes place and the reductional division occurs here, right? And process of formation of mature ovum takes place here in the maturation phase. So we will discuss one by one. Look at this diagram. See, this oogenesis starts in the female even before their birth, right? It starts in their gestational time. When their development takes place, the oogenesis starts, right? So multiplication phase, look at the polarity. All the cells are in the multiplication phase are 2n, means there are only equational division, that is mitosis division is there. No meiotic division is there, right? So after that, second phase is growth phase, in which growth is takes place, increase in the size takes place, and in the third phase, maturation takes place, crossing over is there, and reductional division is there. One cell divide into 4 and 4 into 16. Out of 4 cell form, one is mature ooted, while rest are polar bodies, right? So at the end of oogenesis, what we got from one cell, we got one mature ovum and 3 polar bodies, right? So there are 5 stages of oogenesis. First is the germinal epithelium, cells divide until many diploid oogonia are formed. These diploid oogonia are formed from surface germinal epithelium cells. They divide into primordial germ cells, and these primordial germ cells divide into diploid oogonia. Now, these oogonia grow to form mature oocytes that surround by a layer of follicle cells. The oocyte undergoes the first meiotic division to become a secondary oocyte and first polar body. As I explained you, when ovum formation takes place, one ootid is formed and as well as polar body. The follicle cells surrounding the primary follicle develop into secondary follicle. Now, the follicle layer of the secondary oocyte thicken and fold to form Graafian follicles. These follicular cells are further divided into theca interna and theca externa, right? When the Graafian follicle become mature, it will move towards the surface of the ovary wall and rupture to release the secondary oocyte, right? There is one difference, secondary oocyte and ovum. Secondary oocyte is the incomplete ovum because meiosis 2 is not completed. Meiosis 2 completes only when it makes contact with a sperm. So first we explain 3 steps. First step is multiplication phase. As I explained you, primordial germ cell undergo mitosis. They form oogonia or egg mother cells, and then division stops at the primary oocyte. Now, growth step takes place, in which growth of primary oocyte takes place. This growth phase are consist of pre-vitellogenesis and vitellogenesis. Pre-vitellogenesis is the preparatory stage of vitellogenesis. Basically, what happens in pre-vitellogenesis, it is the nuclear and cytoplasmic substances nuclear as well as cytoplasmic substances, they comes into the action. Whereas in vitellogenesis, yolk formation takes place, right? We will explain each of them one by one. First, as I explain you, pre-vitellogenesis, it consists of growth of both nuclear as well as cytoplasmic substances. First, we explain growth of nuclear substance. Nucleus now become enlarged. Why? Because it is due to the increase in the production of nuclear sap. Basically, a change in the structure takes place. Nuclei might appear bloated and termed as germinal vesicles. Now, chromosome may increase in the length and several loop may transfer, indicating that a massive RNA synthesis for protein production. Lambrusque chromosome. In some of the vertebrates, this giant chromosome is there. It's basically it forms by repeated round of nuclear division. And nucleolus is the germinal vesicles which involved in ribosomal RNA synthesis, right? Now, growth of cytoplasmic substances. Mitochondria, they increase in the number. Once they increase in number, they increase the oxygen consumption because mitochondria are known as powerhouse of the cell. So if their number increase, the power consumption or the oxygen consumption also increases, right? They may comes in the aggregate forms, right, known as mitochondrial cloud, right? Now, Golgi body, they disappear completely into the cortical granules. These cortical granules have function in blocking polyspermy. We will discuss this in later section. Endoplasmic reticulum. They form stacks of double membrane with arranged parallel or in spiral arrangement. Mostly ribosomes are not attached to surface. Membranes are perforated. Pores are there. Cortical granules, they are present inside the membrane of plasma lemma. Plasma lemma is the cell membrane of ovum, right? Spherical or simple membrane. It contains various mucopolysaccharides which synthesizes fertilization membrane during fertilization, right? Vitellogenesis. Vitellogenesis is the process of formation of yolk. Now, yolk formation takes place at two sites. One is on-site, one is off-site. On-site vitellogenesis is known as formation of yolk inside the ovary, right? Whereas off-site vitellogenesis is the formation of yolk in the liver. Yolk is stored form of food. Right? In amphibian and fishes, it happens in mitochondria. This yolk serve as food and nutrition during growth of ovum. Right? Now, physicochemical nature of this yolk. It consists of proteins, phospholipids, fats, and different combination. It may be of two types, proteinous yolk or fatty yolk. Some animals may contain both type of yolk. In avian yolk, let's see the composition of avian yolk. 48.7% water, 32.6% phospholipids and fats, 16.6% proteins, and 15% carbohydrate. Functions of the yolk, as I earlier explained you, it acts as storage of food. It determines the size of egg. See, in human, very less amount of yolk is present. So human egg is microlethal, whereas amphibia reptiles, their egg is macrolethal because huge amount of yolk is present. So this yolk can also influence the differentiation of ooplasm and cleavage. So cleavage also different in case of nature and amount of yolk, right? Influences morphogenetic movement of blastomeres during gastrulation. These are the developmental stages after fertilization. Its amount governs the nature of development of egg. Nutritive cells. In human, two types of nutritive cells are present. One is follicle cells, which surround the ovum, and these follicle cells, these divide into two layers, theca interna and theca externa, where nerve cells provide cytoplasmic bridges and during the growth phase. First, we will discuss about follicle cells. These follicle cells are found in mammals as well as in some vertebrates. They surround oocyte during growth and maturation phase. They are derived from germinal epithelium cells of ovary. Initially, a single layer of follicle cells, later converted into multiple layer, as I explained, theca interna and theca externa. Follicle cells, along with developing oocyte, are known as Graafian follicles. Nerve cells, they are present in addition to follicle cells in some of the invertebrates like in annelids, insect, mollusk also. They derived from egg cells by mitotic division. Nutrients from nerve cell pass to the oocyte, but there is no microvilli, right? Nerve cell might be consumed or engulfed at later stage. See, this is the cytoplasmic bridges formed by nerve cell, and this layer is known as follicle cells layer, and the bridges formed by the nerve cell are known as cytoplasmic bridges. Now, maturation phase. The primary oocyte undergo first meiotic division, means DNA content remains half. Polarity changes from 2n to n. Since there is an unequal division of cytoplasm, one of the cell is known as ovum and another cell is known as polar body. This is known as asymmetrical division. So if one asks you what is the example of asymmetrical division, asymmetrical cell division, so oogenesis is the example, because here One cell contains all the cytoplasmic content, whereas other one is contains no cytoplasmic content. That is now differentiated further into uh, polar bodies, right? Ovulation occurs in secondary oocyte because, as I earlier explained you, secondary oocyte stage because meiosis 2 is not completed. Second part of meiosis takes place only after the contact of sperm, right? The nucleus of secondary oocyte divides again to form another polar body or an ootid. As there is no metamorphosis occurs in ootid, it may be called as ovum. See, again, this is a flow chart. Oogonium converts into primary oocyte and then meiosis takes place. And in meiosis 1, polarity remains half to n to n. Then equational division takes place, results in the formation of one mature ovum and three polar bodies, right? So what is the output of oogenesis? One ovum and three polar bodies, right? So this is the difference in spermatogenesis and oogenesis. The basic difference is that spermatogenesis continues throughout the life once achieved puberty, but oogenesis stops after menopause, right? And second difference is that in oogenesis, from one cell, four cells are formed, out of which one is ovum and three are polar bodies. Whereas in spermatogenesis, from one primary spermatocyte, four mature sperms are formed, which later fuse with each other to form a zygote. Maturation phase. In maturation phase, meiosis results in the formation of one haploid ootid and three haploid polar bodies, or also known as polocyte. It's divided into two phases. One is maturation 1 and one is maturation 2. In maturation 1, meiosis 1 divide diploid primary oocyte into one large and one haploid secondary oocyte and one haploid polar body. In maturation 2, meiosis 2 divides secondary oocyte into one functional ootid and one polar body, that is second. Now, the polar body 1 also divided by meiosis 2 to form polar body 1 and polar body 1A and 1B, polar body or polocytes. Second maturation division normally takes outside the ovary. In some animals, it takes place before the fertilization, and in another, it occurs after fertilization. In many non-chordates, both the maturation 1 and 2 takes place inside the ovary. The process of oogenesis finally forms only one functional ootid, as known as ovum. These are the differences in spermatogenesis and oogenesis. Location entirely into the testis, as well as in case of oogenesis, it is ovary. Meiotic division, equal division of cells. In oogenesis, it is unequal division of cells. Germline epithelium, in case of spermatogenesis, it is involved in gamete production. In oogenesis, it is not involved in the gamete production. Gametes, four gametes are formed in spermatogenesis, and oogenesis, only one gamete is produced. Size, there is one critical difference, that size is reduced in the spermatogenesis, whereas size is increased in case of oogenesis, right? So this is the end of topic oogenesis. Now there are some MCQ questions related to oogenesis. Question number 1. The result of meiosis 1 in female for oogenesis will be result of meiosis 1. What is meiosis 1? That is primary oocyte is the right answer. Which of the following hormone show two peaks during a standard menstrual cycle? So answer of this is estrogen. One before ovulatory phase and one after ovulatory phase. Oogenesis starts at embryonic development stage at the time where 4 million oogonia are found in female fetus. At the time of birth, more than one option. So right answer will be the embryonic developmental stage. Arrested stage called primary oocyte is formed during fetal growth, at the time of birth, at the time of puberty, at menarche. So, arrested primary oocyte is formed during fetal growth. Fluid-filled cavity is a characteristic feature of Graafian follicle, tertiary, secondary, or primary follicle. So, answer will be Graafian follicle. First meiotic division of primary oocyte is finished in Graafian follicle, tertiary follicle, primary, or randomly. It is primary follicle. Which of the following hormone show peaks solely in post-ovulatory phase, means after ovulation? So, which of the hormone? Options are progesterone, estrogen, LH, FSH. So, progesterone is the right answer. So, this is the end of topic. Thank you, everyone.